For today, we will be presenting about effects of different concentration of commercial rooting hormones, also known as CRH, on Partuluca grandiflora, also known as monstrose. Commercial rooting hormones such as auxins are plant growth regulators that promote root development. Auxins are naturally produced by the plant itself but can be manufactured and synthesized for agriculture and horticulture. In this experiment, 4 different CRH concentrations will be used, which are 0.0%, that act as a control, 0.05%, 0.10%, and 0.15%. Each concentration will have 3 replicates, with each replicate containing one approximately 10 cm cutting of the monstrose with 3 to 4 nodes. The cutting will be submerged inside 2 cm of the concentration. Weekly observation of root length and number of roots will be conducted for 4 weeks. The observation lead to 0.15% CRH concentration having more uniform root growth with highest total number of roots. In contrast, 0.05% CRH concentration have slightly longer roots but fewer in numbers. For the introduction, CRH or commercial rooting hormones like auxins are used to stimulate root growth and development. They are most commonly used in plants to increase the plant roots itself. The example of common auxins are indole-3-butyric acid or IBA and naphthalene acetic acid or NAA. Today we will be uncovering about how the different CRH concentration affects the rooting rates and health of the Partuluca grandiflora cuttings, and which concentration is most effective for promoting root development, and comparing it to the control group, which is 0.0% CRH concentration.
Today, I will show you guys how to perform the experiment. Make sure we wear the glove before starting the experiment. We must wear the lab coat in the laboratory. They are the material and apparatus. Firstly, 0.05% commercial rooting hormone will be weighed using the analytical balance. 1 liter distilled water will be added into the beaker. The commercial rooting hormone will be dissolved with a little distilled water. Then, the commercial rooting hormone will be added into the 1 liter of distilled water. The mixture will be stirred until it is completely homogeneous. The commercial rooting hormone that has been prepared will be stored in the bottle labeled 0.05%. The same procedure will be used to prepare the 0.1% and 0.15% concentration of commercial rooting hormone. They are the material and apparatus. Firstly, the healthy Portulaca grandiflora will be selected and taken for this experiment. Each length of Portulaca grandiflora will be measured approximately 10 cm and will be marked using the marker pen. After that, each length of Portulaca grandiflora will be cut using the cutter. Make sure each cutting has at least 3 to 4 nodes for photosynthesis process. The cups will be labeled with labeling sticker 0.05% 1, 0.05% 2, and 0.05% 3. The prepared 0.05% of commercial rooting hormone will be added into each cup. The base of cutting will be dipped about 1 until 2 cm in the commercial rooting hormone. The same step will be used for the different concentration, control 0.1% and 0.15%. 0.15%. Lastly, all the Portulaca grandiflora will be placed under the uniform environmental condition. The root development of Portulaca grandiflora will be observed for 4 weeks. Make sure every week the data of root development will be recorded.
As shown in this graph, we measure the average root length of plants exposed to different concentration of hormone over 4 weeks. The x-axis represents the weeks and the y-axis represents the root length in centimeters. Different colors represent the different hormone concentration. In the first week, the 0.15% concentration exhibits the longest roots. However, in the second week, the 0.05% concentration surpassed the 0.15% group. Overall, the 0.05% concentration consistently outperformed the other groups in terms of root length. A similar bar graph showing the average number of roots for each concentration. We also track the number of roots produced by each concentration, as this graph illustrates the 0.15% concentration consistently outperformed the other groups in terms of root number. The control group had the highest number of roots in the first week. However, the 0.15% concentration quickly took the lead and maintained its position throughout the study. In conclusion, our study demonstrates a significant effect of hormone concentration on both root length and the number of roots produced. The 0.15% concentration consistently produced a higher number of roots over 4 weeks duration, especially on week 4 with average of 10.8 roots. However, the root length was not significantly higher than in control group, with the control having the highest overall average of length followed closely by 0.15% concentration.
The control group functions as the reference point for comparison with the treatment. This pattern indicates the occurrence of natural root development without any exogenous hormonal effects. Nevertheless, the restricted expansion in comparison to the retreated group suggests that the process of root development was slower and less efficient. The slow development suggests that P. grandiflora can naturally produce roots, although the procedure is prolonged and may be limited by its internal hormone concentration or nutritional supply in the experimental conditions. In 0.05% hormone concentration, root length steadily increased until it reached its highest point in week 3. This suggests that this concentration helped roots grow longer than the control. It's possible that the auxin acts as an external stimulus that speeds up the root growth, especially in the middle of the experiment. But the slight decrease in week 4 shows that this function may stop growing or begin to decline after the initial growth. This could be because the plant has reached a saturation point where it no longer responds as strongly to the external stimulus. Still, the 0.05% treatment did perform more effectively than the control throughout the study. This shows that a low concentration is ideal for root growth without causing inhibitory effects. For 0.1% hormone concentration, it is possible that this data indicates that a hormone concentration of 0.1 triggers some early stimulation of root growth. However, it is also possible that this concentration could lead to inhibition and slower growth in later stages. This could be because excessive hormone accumulation has an adverse effect on plant physiology. Several factors can be pointed out for this situation, such as hormonal imbalance, phytotoxicity, and nutrient depletion. Hormonal imbalance refers to the unstable level of hormone in the plants. A buildup of auxin or other growth-regulating chemicals in the rooting hormone may result from a moderate hormone concentration and adversely impact root development. Abnormal root growth pattern and inhibition of root elongation can result from excessive amount of auxin. Moreover, phytotoxicity is defined as a delay of seed germination, inhibition of plant growth, or any adverse effect on plant caused by specific substance, phytotoxins. Prolonged exposure to rooting hormone can be toxic to plant, leading to inhibited growth or the or the development of fragile, undeveloped roots. Lastly, nutrient depletion. Reduced root growth in this treatment group may have result from nutrient limiting by week 4. Also, human errors such as wrong data interpretation, miscalculation, and inconsistent application of rooting hormone can influence these outcoming results. The 0.15 concentration resulted in the most consistent and rapid root growth across all weeks, suggesting that this concentration may be more effective in promoting the development of numerous roots. This indicates that higher oxygen concentration promote higher root proliferation but may not be as effective at promoting elongation. If oxygen concentration increases above an optimal point, it can prevent root elongation. Other than that, the plant may have allocated more concentration to the development of numerous roots instead of extending individual roots. This alteration in resource allocation may be a natural reaction to elevated hormone levels, leading the plant to prioritize the root development over elongation to enhance its capacity for nutrient uptake. To conclude, auxin enhance root elongation at low concentration, but at higher concentration, they can inhibit elongation while promoting the production of lateral or adventitious roots. Along with the study's finding, the 0.15 concentration led to an increase in the number of roots, while the 0.05 concentration improved root length. Ultimately, in later weeks, the plant may have reached a saturation threshold or experience phytotoxicity where the positive effect of the hormone will exceed by the negative effect, including delayed growth or depletion of resource. This is indicated by the lower root growth at 0.1 and 0.15 concentration.
For the conclusion, a total of 4 concentrations of commercial rooting hormone has been tested. From our observation, we found that plant treated with 0.15% concentration of commercial rooting hormone has the highest number of roots, while the control group had longest roots. On the other hand, 0.05% concentration of commercial rooting hormone has the best balance between root numbers and length without negative effects. For plant that was treated with 0.1% concentration of commercial rooting hormone, the roots initially increased in growth but later hindered it, possibly due to hormone build-up. Based on what we discovered, 0.15% concentration of commercial rooting hormone is ideal for root quantity, while 0.05% concentration of commercial rooting hormone offers a balance of root growth and length. The implications that we have obtained from this project, including that rooting hormones can boost root quantity but not significantly affect root length. Excessive usage of hormone can also slow growth, and hormone levels need to be adjusted to optimize root growth. All of these findings have practical applications for improving plant propagation technique. Before I end the presentation, I, as a representative of my team, would like to give 1,000 appreciation towards Prof. Madia Dr. Nur Hayati binti Yusof for all of her guidance and assistance throughout this project to be successful. All the hard work from our team members are also very much appreciated. We are also grateful for the support of the Faculty of Science and Marine Environment. And with that, I end the presentation. Thank you.